First, I would like to start off with thanking a lot of my colleagues, because this Mixoplankton paradigm has been work over the last decade. We have been undertaking with 3 main grants, and there's a lot of people behind this paradigm who's made it happen. So I wish to start off by thanking everyone and all the funding you have received for this. So my research tools, as Denise very kindly explained, they are basically systems biology approaches based. I use these approaches to explore plankton ecophysiology. And in essence, what I basically have, which is unique in the things I do, is that I look at mechanistic feedback and feed-forward controls. So as you heard from Denise, my background is very much biology, ecology, and maths. So I bring all these together to look at how the physiology and how the organisms and things work in ecology. And what was also quite unique when I started in this journey with my PhD was having these feedback processes. So for example, if you eat, the gut gets full and there's a feedback on ingestion rate. So those sorts of things were quite new when I started. So I was told that we've got a very diverse audience, and to start from the start of my journey and work my way to today. So I'll start at the very basic, which because as Denise mentioned, sorry Denise, I keep mentioning it, I started with botany and then went on to ecology, and then, so I'm not a marine biologist, I made my way into marine ecology doing lots of different things. So water plankton was one of the first questions I looked at. These are organisms which drift in the oceans, but cannot swim against water currents and tides. And the really exciting thing about these it's not just the microscopic organisms, things like the larvae of crabs, larvae of sea stars, of sea jellies, they're all planktonic, have got planktonic phases, but sea jellies are plankton as well. And a vast bulk of the marine life comprises of microbial plankton. This is in numbers and also in biomass. And this is what got me hooked into the world of marine systems. So microbial plankton, so that's where most of this talk is going to be in a part of that. These are, again, microscopic, single-celled microscopic organisms, and they we can very, very, very broadly divide them into the heterotrophic bacteria, the phototrophic cyanobacteria, and these two together form the prokaryotes group. And then we have got the single-celled protists. These form the eukaryotes. So in terms of, if you look at the tree of life, the eukaryotes, we are eukaryotes as well. So we're partly related to protists. So the microbial plankton in marine ecology, the traditional view is we have these microbial plankton at the base of a food web, food chain. We've got the phytoplankton, which are making food. And then we have got the microbial zooplankton, which eat these phytoplankton, which are then eaten by the higher zooplankton, protopods, krill, by fish, etc. And they, and all the nutrients, etc., they go and help the phytoplankton then, provide the nutrients for the phytoplankton. So this is very much based on a plant-animal dichotomy, very much like the terrestrial system, where you've got the plants making food and then the animals eating, defecating, regenerating, which helps the plant grow again. So when we look at the microbial protist plankton now, we see that we have got the plant-like phytoplankton. So this is literally like a plant using light energy, carbon dioxide takes up nutrients to make food, releases oxygen, dissolved organics, etc. And the classic ones are the diatoms in this group. And at the other side, the animal side, and these are again the single-cell microbial protist plankton. So these are eukaryotes and single-celled. These are the protozooplankton, which are eating. So they eat prey. They take up oxygen, respires out carbon dioxide, that like us, and also releases nutrients and voids, defecates material. So we have got very much this. This is the in the microbial protist range. We have got the protozooplankton and the phytoplankton, the plant animal. And then the next question, which I asked is in evolutionary terms, which came first? Was it the plant-like phytoplankton? Did you think the plants must be there first, or the animal-like protozooplankton? And this is where it was really exciting for me to find out it was actually the protozooplankton in the Protist kingdom. They were the ancestral form, and the diatoms, the phytoplankton, are more recent forms. So this then led to looking at me researching how this actually happened. It's a lot of work done by John Raven et al. And one of the things which I found, one of the things which is really exciting to find how this happened was, this is the view, was that we had the cyanobacteria, these are the prokaryotes. We had the protozooplankton, the ancestral protists, they ate these cyanobacteria and were digesting. At some point, instead of digesting its prey, it would have retained the plastids, and gradually over time, again at some point, these retained plastids would have gradually incorporated in their own genomes, and they would have become phytoplankton by developing the capability to photosynthesize. So next, the question is, okay, we've got the two phytoplankton, protozooplankton, the ancestral and the more recent forms. How did this happen? Did it happen just like that? Or were there lots of things in between? And this is where our main today's subject turn up, the Mixoplankton. So in essence, this transform or the evolution from animal-like protozooplankton to plant-like phytoplankton would have happened via the Mixoplankton. And what is interesting as well, there's some researchers which show that it is not a unidirectional. This has happened over millennia, and it's gone both ways. We've got it gone from animal to plant and plant to animal. So this is not, not a unique one-time process. So what are Mixoplankton? So these are single-celled planktonic protists. So these are eukaryotes, again, related to us, very, very far away. And they have the ability to acquire nutrition through phototrophy. So it's making food like the plants, and also phagotrophy, eating like the animals. So these are the Mixoplankton. And it's a coin that we termed recently and out of one of the projects we're working on at the moment. So the term is new. Are the organisms new? Are these new discoveries? No, they're not. The popular perception has been that we have got the phytoplankton and we've got the protozooplankton. So we've got these in the microbial marine systems. And in between somewhere, there have been these mixotrophic protists. So they are doing eating and photosynthesizing, whether in the between these two traditional end of spectrum organisms. And what happened though, these mixotrophic protists have been considered traditionally as inferior freaks and some people who are a bit polite curiosities. So they've been sort of on the edges of mainstream research. They've never been considered as important. So this, before we go into why Mixoplankton, I've now given you two different terms. I said Mixoplankton and Mixotroph. So Mixotrophic protists, which if you go into literature before 2019, so it's mostly you'll find words like Mixotrophic protists. And we did have a discussion about what should we call these things. So we decided to go with Mixoplankton. A is to give it the recognition it deserves, just like other plankton, so phytoplankton, zooplankton, bacterioplankton. If you think about it, all these communities have their own terms, so we thought they should have their own terms. And the reason we didn't want to use mixotrophs is because mixotrophs is a much broader definition. So a mixotroph, when we look at the actual definition, it's a combination of autotrophy, so making your food, and heterotrophy. So this is then acquiring energy by ingesting or by taking up other nutrients or food. This autotrophy could be phototrophy, which is using light energy and inorganics to make food. So this is, and the heterotrophy could be, one example of heterotrophy is osmotrophy, where the organisms can use dissolved organics, the sugars, etc. So these are taken up. And this would be mixotrophy. And under this definition of mixotrophy, we would have the cyanobacteria, the prokaryotic cyanobacteria. We also would have the diatoms, which are our traditional phytoplankton. And therefore, when you look at it, this is where the conundrum arose that, okay, if we're going to call these organisms which are eating and photosynthesizing mixotrophs, then how do we differentiate them from diatoms and cyanobacteria, which are technically mixotrophs as well, because they're photosynthesizing and doing osmoheterotrophy. So when we have phagotrophy then, so this is eating, ingesting of a particle which involves killing another organism. So it affects the food web in a very different way because it's removing another organism from the food web. This is where we get our mixoplankton. So in essence, what we have, all mixoplankton are mixotrophs, but all mixotrophs are not mixoplankton because we've got the diatoms of cyanobacteria, which are mixotrophs, but they're obviously not eating. Well, at least nobody's shown them to eat yet. So they're not mixoplankton. And this is the difference. This is why we thought, right, we need to have a term which actually identifies these organisms which have their unique niche in marine ecology. So the next question, of course, we've got mixoplankton, and then I do modeling. So, for me, I wanted to know how many can we, if we're looking in a model system, in a marine ecosystem model, so can I put one box in, calling a mixoplankton, or do I need to put lots of different boxes? So we started with the Protist. We started to put them into functional groups, and the best way, I thought it was just to start with a yes, no. So we started with the Protist, asked the first question: can they fix carbon? So can they photosynthesize? If the answer is no, we got the protozooplankton, or this is the traditional heterotroph. So things like red noctiluca, protoperidinium, they fall within the protozooplankton. If they can fix carbon, the next question we asked was, can they also eat phagocytose? So can they also ingest food, eat? If the answer is no, then we get our phytoplankton, the traditional phytoplankton, which are photosynthesizing but not eating. And this is where we have our diatoms. And of course, we're on the screen, so there is a yes. So now we have got something, a yes, so we've got something which is photosynthesizing and something which is eating. And here we get our mixoplankton. So it would be nice if it was the simplest, but we thought, let's find out how these mixoplankton work. So the question was, okay, it's going to be photosynthesizing. Can it, if you think of it evolutionarily, how they evolved, have they got their own chloroplast? Have they got their own genetic material to photosynthesize? If the answer was yes, we get our what we call the constitutive mixoplankton. So constitutive because it's got the constitutive capability to make to fix carbon, and it also eats. And this, so this is we've got the constitutive mixoplankton, a schematic of it. So it's got its own chloroplast. So it's taking up carbon dioxide using light energy to make food. It's also eating, so it's doing both. But this, because it's got its own genetic material, it can replicate it and it can grow. So it's not dependent on any other organism to make its own chloroplasts. And some of the examples, there's some of the very iconic examples which fall in here. So we've got the coccolithophores, the tripos, serratia, the different dinoflagellates and different cryptophytes. They're all within the constitutive mixoplankton group. And here we've got, thanks to Urban, Dr. Urban Tillman and Avi, a video of, we've got the tripos, which is a constitutive mixoplankton being eaten by another constitutive mixoplankton. So we've got these. You can see it, it's being attacked. So of course, we're talking of microns here under the microscope. These are all single-celled organisms, and this is what's happening. So it's quite exciting, actually. Brutal, but exciting. So you've got these three which have attacked this. It's literally engulfed. So what they do, they literally open up and engulf the cells. There are lots of different ways they eat. Some of them will stick a peduncle like a straw and suck things out, and this is just engulfment. So they're fighting for the last bit. There's clearly a bit they don't like, which they've left, and they've gone on to find the next victim. So we're back to the chart then. We've got, what happened? I think we went. Okay, yeah. So we've got the constitutive mixoplankton which have got their own chloroplasts, and then there are mixoplankton which don't have their own chloroplasts. And these then, the question is, how do they acquire phototrophy? Where do they get the chloroplast from? The question there we had was, do they need specific prey? Do they have to get chloroplast from a very from specific clade of prey? And if the answer is no, we have our generalist constitutive, non-constitutive. So non-constitutive because it doesn't have its own phytoplankton. I'm sorry, its own chloroplasts. So in essence, what we have got, we've got the schematic of a non-constitutive mixoplankton. It's eating, so it's got digestive, it's taking prey in to digestive vacuoles. And then it will have some of the prey where it will retain the plastids from it. So it will take these and keep it as kleptochloroplasts in their cell and use that to photosynthesize. So because it's from generic prey, so they haven't evolved to use very specific prey. The problem they have is they need frequent top-up because they don't have the capability to look after these plastids for very long. The advantage for it is, of course, because it's generic, it is not dependent. So again, depending on the situation it is, it can do very well. And examples of these generalists include some of the really, what I think, the pretty ones. So we got the Lavoia and then the Strombedium. So these are also quite preferred food items in the marine food web. So we're back to the chart. So obviously there are a bunch of mixoplankton which needs very specific prey or acquired phototrophy. So we call them the specialist non-constitutives. And then we had a massive discussion, can we leave it at that? And the discussion led to saying, no, we can't leave it at that, because we've got the specialists which keep plastids, a bit like the generalists, but from specific clades. And we've also got another set of specialists which keep the whole prey as endosymbionts. So again, we're still talking of a single mixoplankton, single-celled species where it's keeping lots of different unicellular organisms as endosymbionts. So with this plastidic specialists, we've got, again, it's taking up the prey, but this time from very specific items, and then it keeps the plastids from it, or sometimes a bit of the nuclear material, and it's also eating on the other hand. What is advantageous for this compared to the generalist, because it's acquiring phototrophy from specific prey, it appears that they have better control of photosystem. So they can go for sometimes days and weeks without needing a top-up. And the examples of the plastidic include the Mesodinium species, the Mesodinium rubrum, there's Mesodinium, and also lots of the different Dinophysis species. They're both plastidic specialists. And it's interesting, there's a very interesting relationship between these two as well. So the Mesodinium rubrum, it requires plastids from teleolax, a cryptophyte, a clade, teleolax clade. So it's going to take these in, keep the plastids from here. The Dinophysis requires the same chloroplasts from the teleolax, but because of the size difference, they can't eat the teleolax. So what they do is the Mesodinium will steal these, and then they will go and rob the Mesodinium of the plastids. So we've got a video from Mangyun Park, and it shows, so we've got here a Dinophysis, and that's a Mesodinium there. Yeah, there we go. Okay, so we can see here, you can see it's captured it. So what the Dinophysis does, it puts a peduncle, a straw-like thing into the Mesodinium as we go through, and it's here, you can see it here. It's literally, there's a peduncle and it's sucking all the guts out of the Mesodinium. So it's, and it literally, that's how it will get its plastids, gets its everything else from it. Exciting organisms. And then we've got the other specialists, which are the endosymbiotic ones. So these organisms, they don't go into any hassle of trying to take the plastids out. They just take the entire prey and keep them as symbionts and use them to do phototrophy. And examples include some of the biogeochemically important, biogeochemistry important organisms. So there's the forams and the radiolarians, the rhizarians. Here's another video by Professor Howard Spiro, and what he's showing is Orbulina, which is a foram, is eating Artemia. So we've got so this is an Artemia. Noclea, these are used a lot in aquaculture, for aquaculture food. This is a foram, this is a central capsule. We've got all the different filaments out of it. It's captured the Artemia and is gradually pulling it in towards the central capsule. And I should have to fast forward this. It goes on for a while. Just gradually pulling it in, and what happens is you'll see soon that Professor Spiro adjusts the focus of the microscope, and you'll find all these yellow things are actually the endosymbionts, and you should see them soon enough. There we go. So you can see these little dots here. So these are the endosymbionts. So what happens again? What happens is these forams of the rhizarians, they have these endosymbionts, and one of the things which has been suggested, so Fabrice Not, who works in Villafranche, he says that these things are sent out. So over in the morning, they will send these endosymbionts out, and then in the evenings, they'll bring them back in. And of course, in the meantime, they're also eating and digesting. So we've got that. So when we go back to now our Protist functional groups, we've got in the we've got our two original ones, traditional ones, the protozooplankton and the protozooplankton and the phytoplankton. So the animal-like protists and the plant-like protists. And then now, instead of just those two, we have got not just one mixoplankton, we've got 4 different functional types of mixoplankton. And this is after about, I think, 4 years of discussions, because we had, on one hand, people wanting to put more and more in because there's so many strains and species, etc. But this is what we agreed to narrow it down to. So the next question is, of course, okay, it's all very well saying, yes, you've got a chart, we've got all these types, but is it really necessary? Again, going back to the modeling aspect, if we're going to introduce something, how many boxes do we need to introduce? Do we need all these different mixoplankton functional types? And to do this, we looked at where do we find them? Does it matter? Do we find, are these, for example, in only one location, or are they across different areas? So we did 2 biogeography studies, one of the constitutive mixoplankton. So these are the ones which have their own capability to photosynthesize and eat. And this is what we found. So on this axis, this is the size. We're going from less than 5 micron to greater than 20 micron. And here we've got the broad taxonomic functional groups. The white spaces in this means that we do not know of any species which fall within the size range for these taxonomic groupings. And the white in the maps itself mean that they're not there, they've not been observed in those areas, but it doesn't mean that people have gone to find them. It means people have not necessarily looked for mixoplankton in all these different Longhurst provinces. And also we found that understandably, that there's a big bias towards dinophytes, dinoflagellates. People are more keen to study rather than a whole lot of the other organisms. Of course, this was done way back in 2017. Since then, we have got another one of the iconic, well-known cosmopolitan species, the Emiliania, the cockleotophores, which have joined the Constitutive Mixoplankton group. So these are the ones which you can see from space, like the milky seas, which Jules Verne talked about. And they are these organisms with the calcium carbonate pockets around, and very important for over millennium. They're the ones which have been in the White Cliffs of Dover in UK. They were responsible for these, and they're even now they're very important organisms in the marine ecosystem. But and it's only recently it's been found. Miguel Prada and Avrami have found that these coccolithophores, Emiliania, are mixoplankton because they eat bacteria as well as photosynthesizing, so they constitute mixoplankton. The other biogeography study we did, of course, was a non-constitutive mixoplankton. So these are the ones which acquire phototrophy. And what this is the GNCM, so the generalists. And we looked at the plastidics and also then the symbionts. And again, we can find that they're absolutely everywhere. Again, the white longhorse provinces mean people haven't found them. The asterisk ones means people have looked, but not necessarily found. But again, it's the very few studies. But this was the most interesting one, was in the symbiotic, because we were not expecting them to be so everywhere all the time. So basically, we came to the conclusion that yes, they are, all the different functional types are everywhere. They do not occur at the same time. They occur at different seasons. So depending on the seasonality, you'll find the different types, whether it's a CM or NCM, within the NCM generalist, plastidics, endosymbiotic, etc. So from that, we concluded that they are all important. That's standing for now. And then, okay, so the next thing is, okay, they're important, but then of course, are they good or bad? How do they actually work? Yes, we can say certain things are good, like some things we know of, but broadly, when we look at the functional groups, whether are they good? So in a healthy ecosystem, where there's no pollution, everything's fine, working well, we have got all this wonderful constitutive mixoplankton, the ehuxle, the tripos, the cryptophytes, and they are important as food for the copepods and the krill, which are then food for fish, the high trophic levels of fish, the cetaceans, etc. So they're obviously important because they're making this link between primary production to the high trophic levels. In an unhealthy ecosystem as well, where we've got pollution or different factors, we've got the dinoflagellates or different, which go and form different blooms. And this is where then we end up with situations where we get the toxic blooms, we get different fish killing blooms, and the closure of shell fisheries. So the dinophysis, while it doesn't affect the shellfish, it will affect the humans by giving them directly shellfish poisoning, any shellfish which eat dinophysis. So in unhealthy ecosystems, they become problematic, especially for us humans, and also in some instances for the other trophic levels. And this is what it can do. This is absolutely amazing video from Peya in Pear's lab. It's called Carlodinium. So we've got a constitutive mixoplankton. You can see these little things here. These are the dinoflagellates, and this is a copepod larvae. And you can see they are actually attacking this copepod. So basically, the Carlodinium eat by engulfing. So they bite into and take chunks off. And you can see it literally goes and attacks this copepod. And this is another video of Teya's, where again, we've got the Carlodinium, which is attacking this poor worm. And again, it's going and starting to eat, and it will eventually, gradually, it's going to kill it and keep eating. So what is interesting with this side of things is that usually we go from small things are eaten by big things, and and move up. But here we've got an inverted trophic pyramid in some ways, where we've got the small things eating big things instead of the big things eating small things. So these are, again, looking at it, trying to think big, like biogeochemistry wide. These are, again, looking under the microscope, a lot of these studies. So looking at what are the implications for the paradigm in marine ecology. So we started to get, in my journey, we started from very small, simple things and going to big things, bigger models. So we looked at the plankton system. So this is a traditional one where you've got the phytoplankton, zooplankton, typical one with the cyanobacteria. And with knowing all the different functional types, we found that the plankton systems is very much more complex than what has been known in the past and acknowledged in the past. And with all the system, then the next thing is, okay, this, especially with the COP26 coming up, there's lots of discussions about climate change models. So one of the things we looked at was what do the climate change models have? The main ones, which are the prominent ones used for the IPCC predictions. And this is what we have, these models have. We've got the nutrients, some of them are just one single. So there's nothing here because this is very much a physical oceanography model. So there's no biology in this. In some of the others, there's single nutrients, dual, multi-nutrients. But if you look at these, this is the phytoplankton and the zooplankton. So that's all there is. So if you think of just the phytoplankton, zooplankton, the previous slide, that's complicated enough. And then when you think of mixoplankton, they're just not there. So this is what the climate change models, the ocean models have at the moment, very much a traditional NPZ structure. But of course, the question is, why do we need to include them? Originally, there are a couple of models like Beth Fulton's in Australia and also Barrett Becker's AirSim, which said, okay, a percentage of phytoplankton, percentage of zooplankton would be mixotrophic. The question is, yeah, can we just go with it, or do we need to include them? So the ecosystem implications, to find out whether it makes any odds, I did lots of different modeling ones, but I'll just look at one of the simple ones and move to the more recent ones, which we've done in putting it in a complex ecosystem model. So we looked at a simple traditional model where we've got the phytoplankton and the microzooplankton. So it's just making food, eating food, dissolved organics, DOM is dissolved organics, material, we've got the bacteria, which is regenerating, it's eaten by the microzooplankton. So this includes a microbial food web of Azam et al. In the 1983. And then we've got the DIM, dissolved inorganics. Then what I did was I replaced the microzooplankton with with the CM, so Constitutive Mixoplankton. So these are the serrations, apocalypts, etc., the ones which have got their own phototrophic capability and also eats. So now we have got these organisms are eating the phytoplankton and also eating the bacteria, but also taking up GIM. Sorry, you can't see them. This arrow is a double-headed one. And the other one I did was replaced this microzooplankton, the traditional one, with the non-constitutive mixoplankton. So these are acquiring phototrophy from the specific prey. So they're taking, stealing the chloroplasts to do phototrophy. So implications, so when I ran these models, the traditional model gave a nice bloom. So we've got on this axis, we've got the biomass in carbon per liter, and there's a day, so 30-day simulation. So we can see this is the green is phytoplankton, so the phytoplankton goes up and down, the bacteria is there, and then you've got the protozooplankton and the microzooplankton, which increases. So this is a very typical spring bloom, autumn bloom scenario. When we replace the Protist with the constitutive mixoplankton now and run the same simulation, well, we've got the constitutive mixoplankton literally take over. So this is very much a scenario where we can have a harmful algal bloom type situation. And when we replace the protozooplankton with a non-constitutive, then we see again, we get a bloom. So it is a bit like the traditional one, but again, we can see that the non-constitutives are much higher biomass and the dynamics are different. So the important thing with these things are it's not only looking at these dynamics which differ, but it will also mean what is being released, what is being regenerated, the different inorganics will be different, and the different biomass available for the high trophic levels are going to be different. So from this, we saw, okay, in a simple case, it was important. So the next one, what happens if you put these mixoplankton in an ecosystem model? So we looked at the European Regional Seas ecosystem model, AirSim, which is based in Plymouth Marine Lab. We had a student, PhD student, Susanna Lele, who did the work on this. And what Susanna did, she took the traditional AirSim model And she replaced all the Protist functional groups, which are known to have Mixoplankton functional types, with these Mixoplankton models. So for example, the flagellates she put in the CM micros. This is also, the AirSim's also quite a nice model to look at, because it also does allometric scaling. So this is, so at the bottom here, you've got a pico, nano, and going up to meso. So Susanna put these Mixoplankton functional types, not just, it was not, it was allometrically scaled as well as functional type. I didn't differentiate it. And then this one then was run in an L4, which is a station of Plymouth. It's here down here. So that's a long-standing station with lots of data. Every week they go and collect data and analyze these. It was run in a system, water column system with Gotham, run for 9 years, and the initial conditions were all configured according to the L4 winter. And there are lots of different runs which was done, and I will show the one which I think is like the summary graph, where we can see this is the depth. So this is the surface, zero surface, and going down. These are the months across this axis, so January to December, and the profile, the two profiles, two depth profiles. And this is what we found with the, when Susanna looked at what happens with a seasonal plankton succession. So the green's phytoplankton, the yellow or NG thing is Mixoplankton, the blue is Protist plankton. And what is interesting here, without going to details, is to look at it changes. So looking at the different season and the different depths, there's a difference in which plankton group is dominating at different times of the year, at different depths. And this was then sort of the, the story. This is where the story kind of ends with starting. Are they, what are they, and are they important? Are the different functional types important? So yes, it makes a difference if we put the different functional types versus just putting one. It makes a difference if we consider a mixoplankton versus not. So this is where, this sort of started off with what is a mixoplankton, and then got us to, yes, we need to consider them in big ecosystem models. So 100 years of marine research, and everything's missed this. There's loads of textbooks, loads of BScs, MScs, PhDs, etc., etc. And this has been missed out, these organisms. And of course, the climate change models, which we're using now, they're still missing these organisms. This blue planet, lots of marine stuff. Marine work is everywhere, and nobody's found the importance of the mixoplankton. And yet 50% of the oxygen that we breathe comes from the microbial plankton, organisms that most people don't even know. And 50% of these are the oxygen producing Protist plankton, which are mixoplankton, which most scientists don't know exist. So it's been quite a journey and quite a task ahead as well. So what do we do now? We need to account for mixoplankton in marine ecology studies. So it's in vivo, in vitro, in silico. A lot of the things hasn't happened is because people have gone to look for a plant-like organism or an animal. If you don't expect a plant to eat or an animal to photosynthesize, you won't find it. So we need to incorporate the mixoplankton in our studies. Research tools, again, are very much, research tools, experimental protocols are very much all focused on either a plant like phytoplankton, animal like zooplankton. So these things are not accounting for. So if you just take a simple experiment, grazing experiments are usually done in the dark, but if you think of a mixoplankton, it will be grazing, but if you give it light, it's going to be photosynthesizing as well. So all these experiments need to be rethought of, whether we need new ones or we need to realign what we know. And all these will then hopefully provide us better understanding of the ecophysiology of these mixoplankton. So what we have found that it's not a simple top-up mechanism, it is very much a synergistic process. So there's a whole lot of work which is done by Paige and Jeong's group in South Korea showing that these mixoplankton grow at a much higher rate when they are working as mixoplankton. So by depriving these of prey, for example, the constitutive mixoplankton, they have a lower growth rate. but it's because they don't have everything they need. And of course, if you think of models, we need some time, the data series, time series plots to help understand how it works in the environment, and not just of the Mixoplankton, because obviously these single-celled organisms are now photosynthesizing and eating, so a knowledge of their prey as well as competitors is important. So yeah, not a lot to do then. So what have we been doing? We have, over the last 4 years, so this started off with the grant from the Leverhulme Trust, which led us to the classification, and then that led to a European Commission funded Marie Curie project, Mixoplankton, Marie Sklodowska-Curie project, Mixoplankton, and that's been running over the last 4 years, finishes end of this month. And in this, we have obviously been doing lots of different experiments, modeling, etc. It goes end to end from molecular through to ecosystem modeling, the scientific papers. The things which I was very keen to have in this project are open access, how to do, what to do. So we've got a range of different open access reports, which we have submitted to the European Commission, which eventually will be published, which are the ones hopefully will help to take this science forward. And going forwards, we have got another grant which is running out of Cardiff at the moment, funded by the ERDF and Welsh Government, which is looking at integrating our understanding from field lab work with modeling and trying to take this forward. So that's where we are, and thank you very much for your attention.